Hi, my name is Elisabeth Budslick, and today I will be presenting my final project. So, are you ready to play? Claw game. Let's pick out this one. Now, my topic is going to be the activation of cannabinoid receptors in breast cancer cells improves osteoblast viability in cancer bone interaction model while reducing breast cancer cell survival and migration. So basically the background here that I'm going to introduce is that the endocannabinoid system is involved in inflammation, bone homeostasis, and bone metabolism. The ECS has already been known to restrict cancer progression and maintain osteoblastic function during bone metastasis. So basically what we're really looking into here is how these cannabinoid receptors are going to affect the cancer bone interaction and help breast cancer cell survival. The basic research question that we're looking at is what are the effects of cannabinoid receptor agonist on the interaction between breast cancer and osteoblast cells? And the hypothesis was that there would be a positive interaction between the cancer and bone cells. And now we got another claw game. We'll take this one. So basically the lab methods that were introduced, it's going to start with a cell culture. Then they purchase the chemicals. Then they're going to prep the controlled media, then go to a cell viability assay, then an apoptosis assay, then a DCFDA cellular ROS detection assay, then a Western blot analysis, a cell migration assay, and a stats analysis. So basically what they're doing in this first cell culture is they're going to be basically preparing the osteoblasts and triple negative breast cancer cells, and they put them into a cell culture so they can get ready for the experiment. They then bought the CB1 and CB2 agonist and the antibodies that were needed for the experiment. Basically, for the prep of the controlled media, they were seeding cells into a 6-well plate. Then they were grown until reaching an 80 to 90% confluence. The cells pre-treatment were treated with an ACEA and GW 405833, or the combination of both of them. They then washed, incubated, harvested, filtered, and stored them. This was all done so they could use them in the future experiments. Then in number 4, they did a cell viability assay. I go into more detail on the assays after this. But basically what they were looking for here is they were looking to find the absorbance ratio between cells treated with compounds and the control. And then they took that ratio and multiplied it by 100. Then in number 5, we have another assay, which I will go into detail. But basically for the short version here, they were analyzing the percentage of cells in healthy, early apoptotic, late apoptic, and necrotic phases. And they used the FASC DIVA. Then 6, another assay, they use the DCFDA cellular ROS detection assay. And what they were looking here is they were basically measuring the fluorescence signal of the DCF using a multi-mode microplate reader. Then they went ahead and did a Western blot analysis. And here they used 20 micrograms samples in the gel. They transferred this into the blotting membrane. They used a 5% BSA. They incubated with primary antibodies and then washed and conjugated with secondary antibodies. The proteins were visualized using an ECL and then exposed to X-ray. The protein band was normalized to beta-actin of each lane in order to target the protein expression and to get a better look at what was going on. Lastly, they did the cell migration assay, which basically they were looking to measure the cell migration by looking at the percentage of wound closure area versus the initial scratch area. And then basically they analyzed everything to find out what was going on. All right, let's look around. I like this one. Oh, click to open. So basically, we're looking at the assays now. So I'm going to start off with, so they first did the cell viability assay, and this is what I'm going to later go into greater detail about what was going on here. This was my chosen experiment, but I did want to give you an overall look at what we were doing throughout the whole thing. So here the cells were seeded into a 96-well plate. They were allowed to attach overnight, and they were treated with the ACEA, singularly, or they were treated with the GW 405833, or they were treated with a combination of the both of those two. The MTT was added to the final concentration in a 100 microliter solution. The optical density was measured at 595 nanometers. Cell viability was based off absorbance ratio between cells treated with the compound and the controls, and then this number was multiplied by 100. They did then a apoptosis assay, and basically here they took MDA and B231 cells, and they were seeded into a 6-well plate. The density was 3.3 to 10 to the fifth power cells per well. They allowed overnight attachment. The cells were then treated once again with either ACEA, GW405833, or a combination of the two of those for the next 48 hours. They then stained the cells with an XNV FITC and propium iodide. The apoptic cells were numbered using an FAC scan flow cytometer, and then the FACS DIVA was used to analyze the percentage of cells in healthy, early, late apoptic, and necrotic phases. So then for the next assay that they did, they went ahead and did the DCFDA cellular ROS detection assay. And basically they were measuring the cellular reactive oxygen species using DCFDA. The MDA MD231 cells were then seeded into a 96 Blackwell plate with a clear bottom at 2.5 10 to the 4 cells per well. And they were grown overnight. They were then treated once again with the ACA or the GW405833, or a combination of the both, for 6 hours. They used a multi-mold microplate reader to measure the fluorescence signal of the DCF. Basically, they were looking at the excitation wavelength at 485 nanometers, and then the emission wavelength at 535 nanometers. So the last assay that they used was the cell migration assay, and this was basically looking, it was a wound healing assay, and it measured cell migration. So the MDA MD31 cells were put into a 24 well plate, density at 4.0 times 10 to the 5th cells per well. They made a wound using a I think 200 microliter pipette tip. They then treated it once again with ACA, ACEA, or the GW405833, or the combination of both. They then compared the images of the wound closure and the initial wound scratch area to calculate the cell migration. And we have another one. I think kind of want that. Ooh. So I am so sorry. So the experimental design that was used for the cell viability assay, they were able to use the MTT colorimetric assay for this. And so basically what they were doing is they went ahead and plated cells into a 96 well plate. They kept the density at 10 to the 4th cells per well. They then allowed overnight adhesion, and then the treatment groups needed to be incubated and treated with either the ACEA, the GW40583, or a combination of the two at 3.16 to 1,000 microliters, and they had a half log increment. On day 2, they changed the media and added the new treatment. They then incubated for another day. Then they added MTT reagent to the final concentration of 0.5 milligrams per milliliter and 1,000 microliter culture. This was done on the 4th day, by the way. They incubated for 2.5 hours at 37 degrees Celsius. At the 5th step, they added 50 microliters of stop solution and mixed thoroughly. Then they used a multi-mode plate reader to read absorbance at 595 nanometers. They were able to calculate the cell viability by looking at the absorbance ratio between cells treated with the compounds and the control, and then multiply times 100. So the controls that were used were basically the cells not treated with ACEA or GW405833. I have like a nice little picture right there of it. Then you have the treated cells. So you could either have it treated with ACEA, treated with GW40583, or it was going to be a combination of the two of them. So this is a nice little graph explaining what was going on. Yeah. So basically, the data showed that viability percentage, and you can see it right here. But the cells not treated with compounds are at about 48%. The ACEA were about 48 also. We saw an increase with those treated with just the GW at about 58%. And then we went right back to about 50% with the combination of the two. Overall, the control was at a 100% viability and the treatment was at 50%. So basically, the results were that the cell viability assay alone, the ACEA and GW405833 acted to suppress the UMR 106 cell viability, and the MDA MB231 cell viability when at a high concentration. The overall experimental results were that the ECS protects against breast cancer bone metastasis and its negative consequence on bone cell survival. CB agonist, specifically CB2, were able to prevent breast cancer-induced osteoblast suppression. This was extremely interesting to me. This was my citation, and I also added a little link below it. I will say that in the future, they want to look greater on, sorry, they're trying to find more data to completely support this and show something greater than just vitro, and they want to look very in vitro and work harder to find more information with this. So this was my PowerPoint, and I want to thank you, and I hope you have a great semester. And if you ever want a fun slideshow, go to Slidesmania.